Hello everyone. I'm Rahul Jain. I'm a postdoctoral fellow at CSR Institute of Himalayan Biosphere Technology, Kuala Lumpur, India. And my topic for today's presentation is Narganisha liquefaces, east endophyte of high altitude plant, imparts cold stress tolerance in Arabidopsis. So before starting, I would like to thank Society for Applied Microbiology for organizing this event on sustainable microbiology, the future of combating climate change. Uh, I would like to give you a brief introduction about what these microbial symbionts are and what these endophytes are. So these symbionts are these are the microorganisms that inhabit the plant tissues, and they are ubiquitous no matter where the plant is growing, from temperate region to tropical region. And these microorganisms combinedly or constitutively they make the plant microbiota, which is along with the genome of the plant are referred as the plant microbiome. So they can colonize in both the tissues, whether it is growing above the ground or below the ground. So either it is root or the shoot part, these microbes can be colonized in all the part tissues, particularly the endophytes. Endophytes are the microbes which spend some part or most of their life cycle inside the plant tissues, and without they do not show any kind of symptoms. So they show an asymptomatic growth, and they have role in the plant development, fitness, as well as in the survival. Before moving to the main topic of today's presentation, I would like to bring your attention to this special review, very interesting review that was published in 2020 by the Aquina Rodriguez Group, and it was published in Ecology Letters that states the functional roles of microbial symbionts in the plant cold tolerance. So you can see they have very clearly emphasized that there are very few studies that particularly targets the role of symbionts or the anti-symbiont in the performance of the plant. So they have clearly stated, based on some meta-analysis, that when the plant is colonized with certain symbionts, the performance of the plant, whether it is the cold stress or it could be the heat stress, it gets increased in comparison to the when plant is free of certain these symbiotic microorganisms. Similar is the case. Their tolerance to broad temperature it get increased. It gets increased when the plant is inoculated or it is colonized with some kind of symbiont. So that those symbionts which are beneficial, that is still a major research that we have to find out, and our research also targets that. So for our work, what we choose Arnebia euchroma. Arnebia euchroma, it is a plant that grows in the cold desert Himalayas at an altitude of approximately 4,400 meter above mean level. So above mean sea level. So overall, the condition over there are very deserted, very extremes, and that is why these plants are referred as extremophytes. So why, what kind of extreme conditions could be? First of all, I will start with obviously there are very low temperature conditions, so cold conditions are there. And apart from that, there is a high irradiation. So because of these two situations, you can see there is a large diurnal variation in the daily temperature at such altitude. Besides the temperature, there is low water availability because there is very less rainfall. There is directly snowfall, and then because of this situation, there is also a poor quality, and that is why these are known as cold desert soils. So for our work, we used this methodology. Started with the isolation and characterization of endophytes. So we collected this plant. With the help of conventional microbiological techniques, we isolated these endophytes, and then we characterized them for some plant growth promotion activities. And later on, we performed the plant microbioassays with some potential isolates. So in this study, I'll be mainly focusing on single yeast that we isolated, and after performing these plant microbioassays under cold stress, we studied their plant microbe interaction through expression-based or microscopic techniques. Moving to the next, the first of all, the diversity of this Arnebia euchroma. As you can see, we got a huge diversity of bacteria that was dominated by the proteobacteria, especially while in case of fungi, it was dominated by the Ascomycota. So we got total of 60 bacteria, 24 fungi, and 9 yeast out of it. And Penicillium was the most abundant or the dominated genera in case of fungus, and Pseudomonas was the most abundant genera in case of bacterial isolate. So we also got some of the yeast isolates, interestingly, and that also included this Naganisha that we are going to study very soon, that we are going to I'm going to present very soon. So this particular yeast was common in both, including leaf tissue and the root tissues. Similarly, in case of bacteria, if we'll observe Pseudomonas, Serratia, Brevibacterium, these were the common bacteria which were ubiquitous in both plant tissues, including root as well as the above ground tissues or the shoot. We also studied their plant growth promoting features because the initial for initial characterization, this is very important to know what function exactly they were playing inside the root tissues, right? So we studied the phosphate solubilization, IA production, Sidro4. So surprisingly, we got a very high phosphate solubilizing abilities in the endophytes of the Sarnebi euchroma, whether it is leaf or root, although it was higher in the leaf. Similarly, in case of fungi, it was higher in case of leaf endophytes, lower in case of root endophytes. But the overall ability was quite higher in comparison to the normal plant microbiota that we have observed till date. Similarly, for IA production, we got higher producing IA microorganism in root tissues in comparison to leaf tissues because of probably because of their more function in the root. Similarly, the citrophores were citrophores in case of bacteria, they were more abundant in case of root, while the ability of these microbes in or these fungal endophytes in root and leaf was quite similar. So we all, because of the cold condition over there or the stress condition over there, we also check the production of ACG deaminase by these microorganisms. So we got a quite good number of microbes, including bacteria and fungi, that could produce ACD aminase, and that has a function in the breakdown of ACC, so that the ethanol cannot ethylene cannot be produced and the plant's enzymes can be deleted. Besides it, we also observed the potassium solubilization by these bacteria. So overall, there was a good consortium of bacteria of these microorganisms that could show all these features. So next, that was the major aspect of today's presentation. That is the Naganicia liquefacens strain ARY7 that we isolated this specific strain that we isolated from the root issues of this Arnibia euchroma plant. This belongs to the phylum Basidiomycota and the class Tremellomycetes. And surprisingly, this yeast, is been previously reported from the deep sea environment or the deep sea Japan trench, and that is why it is also known as deep sea yeast. So it was recently, its genome was recently published in Han et al. 20 by published by Han et al. 2020. So this particular yeast we are reporting for the first time as a plant endophyte, and during the characterization study we observed that this yeast can solubilize phosphorus, can solubilize potassium, and besides this it can also produce citrophore as well as the indole acetic acid or the IAA production. So these are the morphological features of this and the phylogenetic analysis. Coming to the major findings of our work or this work, that when we inoculated this particular yeast through the root in the Arabidopsis system, what we observed at low temperature conditions, it ameliorated or enhanced the tolerance of the plant. As you can see through the morphological features simply, that at 22 degrees Celsius as well as low temperature, when the plant was uninoculated or mock inoculated with the was mock inoculated, it showed a reduced growth at low temperature. Okay, while the growth at 22 degrees Celsius was quite good. However, when the plant was inoculated with this ARY7, so at 22 degrees Celsius, the plant showed a reduced growth or a distorted growth. While at low temperature, you can see the plant showed a significantly high growth with a quite high biomass. So even the biomass, you can see the fresh shoot as well as the fresh root biomass was increased in case of ARY7 inoculated plant at low temperature, and similar was the case with the dry weight biomass. Next, because at low temperature there are mechanisms that or there are metabolites that can enhance the cold tolerance either by overproduction. So we checked whether such metabolites or the cold stress responsive metabolites such as proline or NDA, they were high in the AY7 inoculated plant or not. So surprisingly, at 22 degrees Celsius, we observed their amount or the quantity of production was high. But At low temperature, where in the case of ARY7 inoculated plant, the overall proline content and the MDA content was quite low. So surprisingly, that was a fascinating result for us. That why the plant is not producing high amount of proline or MDA in response to the cold stress. So next also electrolyte leakage was also observed low. Electrolyte leakage, what it indicates, that the plants are less, the cells are less damaged, and that is why the leakage of electrolytes in the solution or in the water will be should be less if the plant is intact or the plant cell is intact. However, if it is damaged, then it should be high. But here also we got a very low electrolyte leakage in case of plants that were inoculated with the LT. Then talking about the plant performance or the plant physiological parameters, the chlorophyll content was high in both ARY7 and inoculated plant, and similar was the case for the FVFM. FVFM is nothing but the chlorophyll fluorescence. That actually indicates how active these reaction centers are there in the leaf, so that they can harvest more light and they can do more photosynthesis in the leaves. So based on this, we can say that these metabolites are less produced at plants is maybe it is sensing less cold as compared to the plants that are not inoculated, right? So similar based on the performance parameters, we can say the plant is growing quite well. So next, check what are the generation of ROS. Because of the stress condition, there is every chance that plant will produce more amount of ROS in the leaves because that is the most exposed part. So as you can see at the 20 degrees Celsius in the normal growth, there's no or very less ROS production, while in the plant that were inoculated, but at 20 degrees Celsius that showed a high amount of ROS, while the trend was quite inverse at low temperature conditions. So in comparison, in control, there was a high amount of ROS production by ARY7 inoculated plant. There was a no or very less production of ROS that we quantified using the images software, and here is the result. So obviously the plant is facing less, it is not sensing the cold, and that is why it is not responding on this cold. So we got a very less production of this ROS molecule. And similarly, the SOD expression gene that regulates the ROS generation in the plant tissue, that the expression of this SOD1 gene or the Arabidopsis SOD1 gene was quite low in the plant that were inoculated with the ARY7. So that indicated that plant is experiencing less amount of ROS, less amount of stress in the leaf. So moving to the next part, that is the hormone signaling, because all these stress parameters and all these are much more regulated by the different hormones produced by the plant, or maybe through their symbiotic partner also. So we checked whether ARY7 can modulate the oxygen signaling in the root, because it is also an IAA producer. So as you can see, we used first of all DR5 GFP lines. These are oxygen sensor lines in the Arabidopsis thaliana, and we inoculated these lines with the AY7 and grow it at low temperature conditions besides the normal conditions also. So as you can see, at the normal temperature in the control plants, plants showed a good response to oxygen. However, its expression was quite low in the primary root meristem, but high in lateral root meristems. While at normal temperature, in the case of ARY7 inoculated plants, plant produces very less or no IA or the oxygens. So that indicated that ARY7 inoculation at at normal temperature is somehow inhibiting the growth of plant. However, as you can see in the control plant at low temperature, there was a quite similar trend as in case of ARY7 at 22 degrees Celsius, right? But the plants when inoculated at grown at low temperature and inoculated with ARY7 showed a very high oxygen response. So that indicated that somehow auxins either produced by the bacteria or the end or the auxin produced by plant itself is overexpressing and thereby enhancing the plant's cold tolerance. So that was one evidence that supported that there is some mechanism or there's some auxin-based modulation of cold stress tolerance. Then we check for some of the cold stress responsive genes that are generally produced or overexpressed during during the cold stress. So as you can see, generally if you look at the low temperature bars here, So what you will observe that the expression of all these low temperature responsive gene or the cold stress responsive gene was quite low in case of plant that were inoculated with ARY7. So as you can see, CBF2, CBF3, COR414, COR15B, RD29A, these are all the major pathway genes that regulate the cold stress. Similarly, the LF invertase, FZF, or the zinc finger protein. So all these genes were downregulated, except the FAT2. FAT2 is is a response shown by plant when it experiences stress, particularly cold, because it helps in the fatty acid desaturation. And because of the desaturation, plant cells can accumulate more desaturated fatty acids and prevent itself from different kind of stress, particularly the cold conditions. So that was another. So these were some more evidences that this ARY7 inoculation is reducing the plant's cold tolerance, and that is why the plant is not responding to the cold, is not expressing the cold stress responsive genes. So, because as you could see that at 22 degrees Celsius, the plant that was inoculated with ARY7 showed a slightly uh, unhealthier response, right? So we checked whether there is any biotic stress on the plant or not. So surprisingly, that was not. So SIT2 and PBS3, the first two genes, A or B, that you can see, these are the major salicylic acid biosynthesis gene, and these are produced in response to some of the defense or in case of stress responses also. So SIT2 and PBS3 having role in the accumulation of salicylic acid, they were highly upregulated, more than fivefold or sixfold, I would say. So this suggested that salicylic acid is also produced in response to the AY7 at low temperature. But because salicylic acid is not just involved in the cold stress, but it has also role in defense. So we checked whether the salicylic acid is further modulating any pathway genes that are involved in defense, particularly NPR1, PR1, and PR2. These are all pathogenesis related genes, and surprisingly, their expression was quite downregulated, both all NPR1, PR1, as well as PR2. So this suggested though a salicylic acid is produced, but it is not further activating the systematic acquired resistance or the genes that involved in the pathogenesis, or the defense regulation. So somehow, so this clearly suggested that salicylic acid is having a role in the cold acclimation of the Arabidopsis. So another gene that plays a role in the ABA signaling, that is the RAB18. It encodes a ABA or drought-induced glycine-rich proteins, dehydrin protein. So these ABA are all produced during the abiotic stress, and cold is not an exception to that. And in these conditions, and in response to the ARY7, the plant produced more RAB18, or the plant expressed more RAB18, so thereby showing a role of ABA also in this whole process. Another gene is P5CS1. It encodes a rate-limiting enzyme in the synthesis of proline, and as I previously said, the proline was lesser produced, and so is the expression of this particular gene was reduced at low temperature. While in case of EIN2 is a ethylene responsive gene. It codes for ethylene insensitive factor 2, and it has also people have shown that researchers have shown that its role in abiotic stress. So, but this EIN2 or the ethylene, it combines with the ABA and plays a cross, and it has a crosstalk features with the ABA, and therefore it may have a role in the cold stress acclimation. Another is the PCAP2. This PCAP2, it has a role in the leaf senescence, and its decreased expressions at low temperature clearly says that plants are quite healthier. They are not going to get into the senescence phase. So that was the overall story, and based on all these conclusions or the evidence, we can conclude that this particular Naganisha lactofacins AY7 is to overall enhance the plant performance, particularly at the low temperature. So that was clearly indicated by the high chlorophyll content or the high FDFM ratio. So next is the AY7 regulated oxygen signaling. So because AY7 itself is a IAA producer, so there is a possibility that exogenous production of the IAA by AY7, it modulated the oxygen level in the roots of this Arabidopsis and therefore mediated the cold stress. Next is the high salicylic acid expression in response to AY7 without affecting the reference signature genes can be suggested to enhance the SA-mediated cold accumulation, and the AY7-mediated upregulation of RABT that has a role in the ABA signaling may also contribute to the cold acclimation along with the ethylene-insensitive factor 2, EIN2. So overall, we can conclude that endophytes of such plants or the extremophytes, they can explore micro-mediated extent tolerance in the plants of agriculture as well as in the medicinal value for their sustainable cultivation in the low temperature environment. So I would, at the end, I would say that in line with the theme of this event, that is the sustaining sustainable microbiology, the future of combating climate change. We can say that such kind of studies they can be further applied in the field, so to get climate resilient crop in the near future, particularly like in our region or in the Himalayan region, there are cold conditions, and the crops can be the crop biosphere can be manipulated with such microbes, and high growth or high yield can be achieved. So in the end, I would like to acknowledge CSR ISBT, where I'm working. This is a beautiful campus of our CSR ISBT in the Himachal Pradesh district of India. I would like to thank CSIR, Council of Scientific and Industrial Research, for providing me this fellowship, and in the end, the SFAM, Society for Applied Microbiology, for providing me this opportunity to present my work at such an international platform. And that's my mentor in the INSET, and that is our whole plant molecular biology lab. And we are very much diversified. We work on enzymes, we work on plant physiology, we work on in transgenic, we work in the field of microbiology also. So we got a lot of support from all of our colleagues. And then, thank you for your attention.